Welcome to the Swift Health and Safety Lab. I'm going to take you on a clockwise tour around the lab and show you the standard equipment that you would find in a first year undergraduate biological sciences lab, and also explain a little bit about some of the functionality of some of the items that we've got here in this health and safety lab. This lab, as you join me on the tour, you will see is set out just as you would probably expect any other laboratory. And as we start walking, you'll see there's a first aid kit on the wall and an eye bath, and also the first sinks you come across, which are hand washing sinks. Now you can actually wash your hands in these sinks if you use your right mouse click. You'll be able to wash your hands, and then to stop washing your hands you stand up and the taps switch on and off. Normally students would wash their hands at the end of their experiment after they have taken off their lab gloves. And these sinks are not to be confused with the next sink that we see, which is actually a laboratory type sink where they have three types of taps. There's a hot, a cold, and what we call a grey line, which is the one with the green top. And the grey line tap contains distilled water, which is purified so that students can use this in experiments. And also can rinse equipment with it to make sure it's clean. Hands aren't washed in these sinks, for the main reason being that solutions are poured down the sinks, and therefore things like the taps may well be contaminated by gloves that have touched them. So you wouldn't wash hands in these sinks. And you can also see there's fire blankets and things here, and equipment to be washed on the side of the sink, as would normally be in any lab, and also autoclave disposal bags present at most stations. These are the boxes that you can see as you go around the lab standing on the floor. They are for throwing away things like pipette tips, petri dishes, and other disposables that need to be cleaned and autoclaved before you would actually be able to throw them in the normal waste. Things that have been used with bacteria and stuff like that. This funny little box that you come to on the first item on the bench, this is actually a distilled water producer. So although the taps contain distilled water, this water from here, this ELGO unit, actually contains purified distilled water, and this is the water that you would use in things like PCR reactions and stuff. And then next we have things like pipette tips and micropipettes. You'll see with each piece of equipment there's a yellow tab by them. If you click on these tabs, you will actually collect a note card, and in the note cards is information about how to use the equipment, and also safe operating procedures and any health and safety aspects for each piece of equipment. So next we come to there's microscopes. Now there's two microscopes set up, and as you'll see beside each there are a box of slides. You can switch the power on and off by clicking on the buttons, and you can focus them by pressing the focus button, and this makes the image sharper if it's quite blurry. You can also change what you see under the microscope simply by clicking on any of the slides that are located in the trays to the left of the microscopes. So you can click on the different ones and it'll change the image on the screen beside the microscope. Now in the first microscope primarily there are fungal spores, and in the second microscope we actually have some more fungal spores, but also Drosophila melanogaster, which is a fruit fly. Carrying on round, we have a Bunsen burner. And again, if you click on the yellow tab here, you'll get information about safe use and operation of a Bunsen burner. This Bunsen burner you can switch on by touching the yellow tap at the back. You can change the colour of the flame from yellow to blue by clicking on the collar at the base of the Bunsen burner, and back to yellow by clicking on it again. And beside it we have a little bottle. In that bottle is ethanol, which is a very combustible material. Now if you actually move the ethanol, click the ethanol towards the Bunsen burner, you'll see what actually happens. You'll get well, I won't spoil the fun for you. To stop it, you just move the ethanol away from the Bunsen burner, and don't forget to turn the Bunsen burner off before you move on. Carrying on round, we have a very standard shelving display of what you would see in most laboratories, with lots and lots of solutions and chemicals, and this is standard depending on what type of lab you're in, different chemicals, but usually there will be chemicals and solutions. We go past another sink for lab solutions, and then we come to 2 centrifuges. Now you would need to use your camera controls here to actually look down on them from the top, but if you do look in them, you'll see that one centrifuge is actually perfectly balanced and the other isn't. And when we use this lab for our students, we ask them some questions about the lab, and we ask them which one was balanced and which one wasn't. And then carrying on round, we have a micro centrifuge, one open, one closed. I forgot to mention with the centrifuges, you can actually open and close the lids on those. And again, the balancing aspect of the exercise is exactly the same for the microcentrifuge as with the centrifuge. Then we come to a vortex, which is used for mixing solutions like in Eppendorfs, like DNA and other solutions. It sort of vibrates. Then we have a water bath, again a standard piece of equipment that first year biological student undergraduates would probably come across in their laboratories. And again with safe operating procedures in the yellow tab on the notepad. And then we see 2 different types of PCR thermocycler. There's a big black one which has got 4 blocks on it, so it's a tetrad, and then you can actually amplify 4 plates of DNA at the same time. Or there's the smaller cream coloured box, which is also a PCR thermocycler but just contains enough for 1 block of plates, so it takes up an awful lot less power. Next to the PCR machines, or the thermocyclers, we have a gel power pack and a gel tank. These are used for running the DNA that you may have amplified in the PCR tetrads or thermocyclers. You would then load that DNA onto a gel, place the gel in the gel tank, hook it up to the gel power pack, and then you would actually be able to see the DNA. As the dye front moved forward, you would know that the DNA would be moving forward. Of course, using this type of equipment, you would normally stain the DNA with ethidium bromide, and ethidium bromide is a mutagen. So as you can see, there's a big yellow box next called contaminated waste, and this is where you would place things like contaminated gels, any cloths or paper tissues that you'd used, and any gloves that you'd used that had handled ethidium bromide. Up above, on top of the desk, there's a UV transilluminator. That's what we would use in a lab to actually view the DNA bands on your gel. So it actually shuts the door, shines a UV light onto the gel. The UV causes the ethidium bromide, which has bound to the DNA, to fluoresce, and what you see on the little computer monitor next to it is a typical type of view that you would see on the gel. So then we have more equipment washing sinks. We have an ice and an ice machine, standard equipment in most labs. Certainly if you're doing anything to do with DNA or bacteria, you would want to keep things on ice, and these are found in many labs. And then we come across a fridge, which is at 2 degrees centigrade, and an ordinary type of freezer, but probably down to -17 or -20. You wouldn't store DNA long term in an ordinary freezer because it would actually begin to degrade. And then carrying on the tour, we have a fire extinguisher, we have our health and safety notices, and also on the wall we have our little monitors. One is the fire one, of course, that you would break in case of a fire, but the little yellow one beside it is actually to monitor the carbon monoxide and CO2 in the air, and alarms would be set off, obviously, as these are noxious gases. And then we come back to the area where the gloves and the lab coats are kept, and when students do a tour of the lab, they would usually come and take their coats off at this point, the lab coats, and take their gloves off, and then make their way to the hand washing sinks to wash their hands before they then left the labs, knowing that they'd left anything that was contaminated behind in the laboratory, and their hands were clean and ready to collect their coats, which they would leave outside of the lab on the tables and desks just outside. So that's a tour, a very quick tour of the Health and Safety Lab here at the University of Leicester SWIFT project. You may have noticed that there's a big image in the middle of the floor. If you have time and want to have a look, it will also give you a note card, and if you want to have a look at that note card and read about it, you might find that it's quite an interesting little image we've placed there. Anyway, I hope you've enjoyed the tour, and if there are any questions, please do contact us or have a look on the University of Leicester Swift.